Clarity Clinical Solutions.
How do we know if a treatment really works? There are two fundamentally different ways to find out. The first is the randomized controlled trial, the gold standard of medical evidence. The second is real-world evidence, data from everyday clinical practice. In this video, we will compare these two approaches and explain why both matter for modern medicine. A randomized controlled trial is an experiment where patients are randomly assigned to receive either the new treatment or a control, usually a placebo or the current standard of care. Randomization ensures that the treatment and control groups are as similar as possible, except for the drug being tested. This allows researchers to isolate the true effect of the treatment. RCTs answer the question, can this intervention work under ideal conditions? RCTs are the gold standard for three reasons. First, randomization eliminates selection bias. The groups are comparable at the start. Second, strict protocols and blinding prevent doctors and patients from influencing the results. And third, pre-planned sample sizes give RCTs the statistical power to detect real treatment effects. This is why regulatory agencies like the FDA and EMA require RCT data before approving a new drug. But RCTs have real limitations. They are extremely expensive. A single phase 3 trial can cost $20 to $100 million. They are slow, recruiting thousands of patients takes years. And the biggest problem, RCT populations are highly selected. Strict eligibility criteria exclude 80 to 90% of real-world patients, including the elderly, children, and people with multiple health conditions. The results may not apply to the patients who will actually receive the drug. Real-world evidence comes from data collected during routine clinical care, not from a controlled experiment. The raw material is called real-world data, and it includes electronic health records, insurance claims, data from wearable devices, and patient registries. When this data is analyzed with rigorous scientific methods, it becomes real-world evidence. RWE answers a different question: does this treatment work in real clinical practice? Real-world data comes from many sources. Electronic health records from hospitals and clinics contain diagnoses, medications, and lab results. Insurance claims track procedures, prescriptions, and hospitalizations over time. Wearable devices like smartwatches and fitness trackers provide continuous data on heart rate, activity, and sleep. Patient registries follow people with specific diseases for years or decades. And biobanks like the UK Biobank link genetic data with long-term health outcomes. Let us compare RCTs and RWE side by side. RCTs are conducted in a controlled setting with a highly selected population and random treatment assignment. They can be double-blind and have high data quality, but they are slow and extremely expensive. RWE, on the other hand, reflects real clinical practice with a broad, representative population. Treatment is chosen by the doctor and patient, not randomized. Data quality varies because it was collected for clinical care, not research. But RWE is faster and cheaper, and it tells us about effectiveness in the real world. Real-world evidence excels in several areas where RCTs cannot help. For rare diseases, there may only be 100 patients in the world, not enough for a traditional trial. For pregnant women and children, who are often excluded from RCTs, RWE provides the only safety data available. For long-term safety, RWE can track patients for decades and detect rare side effects that pre-market trials missed. And for comparing two marketed drugs, RWE from insurance claims can answer questions that no drug company will pay to study. But RWE has serious limitations. The biggest threat is confounding by indication. Sicker patients tend to get different treatments, so the treatment and control groups are not comparable. Data quality is another major issue. Electronic health records are full of missing values, coding errors, and inconsistent measurements. Selection bias means patients who receive a treatment are systematically different from those who do not. And most importantly, association does not equal causation. Proving cause and effect from observational data is inherently difficult. The FDA has embraced real-world evidence over the past decade. In 2016, the 21st Century Cures Act required the agency to develop a framework for evaluating RWE in regulatory decisions. In 2018, the FDA published its RWE program guidance. In 2021, Project 351 began piloting the use of RWE for approving new indications of already approved drugs. And in 2023, the FDA released updated draft guidance on considerations for RWE in regulatory submissions. The trend is clear: RWE is becoming an accepted part of the regulatory toolkit. The 21st Century Cures Act was a landmark piece of legislation passed in 2016 with strong bipartisan support. It explicitly defined real-world evidence and real-world data for the first time in federal law. The act required the FDA to create guidance on when RWE is sufficient to support approval of new drug indications. It also accelerated the use of patient experience data and streamlined the development of drugs for rare diseases. The Cures Act recognized that RCTs and RWE are complementary sources of evidence, not competing ones. So which is better, an RCT or RWE? The answer depends on the question. RCTs offer the highest internal validity. They are the best tool for establishing cause and effect. If you want to know whether a drug works, the RCT is your answer. But RCTs have poor external validity. The results may not apply to the diverse patients seen in real clinics. RWE has the opposite profile: lower internal validity but high external validity. The two approaches are not in competition. They answer different questions and work best when used together. The future of clinical evidence is hybrid. Pragmatic RCTs embed randomization into routine care, combining the causal power of randomization with the real-world data collection of routine practice. Target trial emulation designs RWE studies that explicitly mimic a hypothetical randomized trial. And artificial intelligence is improving RWE analysis through better confounder selection, propensity score estimation, and causal inference methods like double machine learning. The future is not RCT or RWE, it is RCT plus RWE working together. Here is what we learned. RCTs tell us whether a treatment can work under ideal conditions. They establish efficacy. RWE tells us whether a treatment actually works in real clinical practice. It measures effectiveness. RCTs are the gold standard for regulatory approval, but they are expensive, slow, and narrow. RWE is faster, cheaper, and more representative, but it struggles with confounding and causal inference. The smartest approach is not choosing one over the other, it is using each approach for what it does best.